How does the protease cascade work in the complement system? Imagine your immune system as a well-organized team ready to fight off invaders. One of its most efficient tools is the protease cascade, a series of enzyme reactions that activate the complement system to protect your body. This cascade is like a chain reaction where one step triggers the next, amplifying the immune response quickly and precisely. It all starts when the body detects something foreign, like bacteria or viruses. The complement system can be activated in three ways: through the classical pathway, the lectin pathway, and the alternative pathway. Each pathway begins differently but converges on forming a key enzyme complex called the C3 convertase. In the classical pathway, recognition molecules like C1q latch onto antibodies attached to pathogens, which then activate enzymes called C1r and C1s. In the lectin pathway, pattern recognition molecules such as mannose-binding lectin bind to sugar structures on microbes, activating enzymes called mannose-binding lectin-associated serine proteases, mainly MASP2. The alternative pathway is a bit different. It starts spontaneously when C3 proteins in plasma undergo low-level hydrolysis, creating C3H2O. This binds to factor B, which is then cleaved by factor D2 from the initial C3 convertase for this pathway. Once these initial steps are set, the proteases cleave other complement proteins like C4 and C2 in the classical and lectin pathways, or factor B in the alternative pathway, to form the C3 convertase complexes. These complexes are like molecular scissors that cut the C3 protein into two parts, C3A and C3B. C3b then sticks tightly to the surface of the pathogen, tagging it for destruction, a process called opsonization. C3b also combines with the C3 convertase to form a C5 convertase, which then cleaves C5 into C5a and C5b. The small fragments, C3a and C5a, act as signals that call immune cells to the site of infection and promote inflammation. C5b takes the lead in assembling the membrane attack complex, a group of proteins that form pores in the pathogen's membrane, causing it to burst open. Throughout this process, the proteases stay attached to the pathogen surface, ensuring that activation stays localized and does not damage your body's own tissues. The entire cascade is tightly controlled by regulatory proteins that prevent overactivation, which could harm healthy cells. This protease cascade is a fast and targeted way for your immune system to identify, mark, and destroy harmful microbes, making it a vital part of your innate immune defenses and a bridge to adaptive immunity.